Hi dear students, a warm good morning to one and all of you. All these days we have been discussing recombinant DNA technology, or shortly speaking rDNA technology or genetic engineering. And the main purpose of we conducting our DNA technology, as we all know, is for our own benefit to produce recombinants for our own benefit, for our own needs. It would aid in the production of pharmaceuticals sometimes, or sometimes certain enzymes, or certain other various other microbial products. Now, whatever be the reason of recombinant DNA technology, the basic steps that we use in our DNA technology is just the same. We take a gene of interest from a particular source, and at the same time we take a vector and we slice the gene of interest as well as the vector using a restriction enzyme, and we combine them together using an enzyme ligase so that they form a recombinant DNA. And now the next step that which we have, once a recombinant DNA is being formed, is to transfer this recombinant DNA into a particular host cell. Now, if you're going to talk about microbial genetics, the most commonly used host is E. coli. But at the same time, you could also have various other hosts. Sometimes you can have a eukaryote such as what yeast, or sometimes even you might have reasons where you have to transfer the recombinant DNA into plant cells or mammalian cells, uh, and sometimes into certain viral cells also. So depending upon what host cell you have different gene transfer methods. And in this session, we will be talking about the gene transfer methods at a whole. If we're going to consider E. coli, as I told you in the previous classes, we have techniques such as transformation and all that. But I'm just trying to give you an overview of the general gene transfer methods where the recombinant DNA is being transferred into a host cell. The host cell, as we mentioned here, it could be an E. coli or a bacterial cell, or it could be a fungal cell, or it could be a plant cell, or it could be an animal cell, depending upon what you are working on. So let's explore it. When you intend to put a gene of interest into a host cell, sometimes you might have produced a recombinant DNA using a vector, or sometimes you might have just taken some naked DNA and you would try to put it into a host cell by vectorless methods. So depending upon whether you use a vector or not, the gene transfer techniques can be named as vector-based methods as well as vectorless methods. If you use a vector such as a plasmid, phage vector, cosmid, phage mid, etc. for gene transfer or to produce a recombinant DNA, it is called a vector-based method. And if you just do not use a vector, or if you just want to directly transfer some DNA into a host cell, then that would be a vectorless or a direct gene transfer method. And these vectorless methods, they mainly involve taking a cell and taking some DNA and transferring it by some chemical methods or by microinjection methods, electroporation methods, or liposome mediated methods. Now, these are all different methods where you're not involving a vector. So don't get confused. You have now, as far as we have vectors, we have recombinant DNA. That's what we have discussed so far. So if you intend to transfer your gene using a vector, that is called a vector-based method. And if you do not use a vector and you want to transfer a gene of interest into something else, that is called a vectorless method. Now, I'm going to talk about firstly about the vector-based gene transfer methods. So, at a whole, right now in genetic engineering, based upon our previous lectures, we have a what? A recombinant DNA. That is vector based also. Now, this vector based recombinant DNA has to put into the host cell. That can be done by different methods. Now, if the host cell is a bacteria, it can undergo the process of transformation. Or if it is between two different bacterial cells, the gene transfer method of conjugation can be used. Or as I mentioned in the previous classes, if you have a tougher cell, or when the cells are not being undergoing the normal calcium chloride treatment method, you use certain techniques such as electroporation so that the cells would be exposed to electric waves and they become permeable to naked DNA. Or sometimes the recombinant DNA can be transferred into the host cell by using viruses, and that involves the process of transduction. So the first four methods here which I have mentioned, they can be used for what? The transfer of vectors. Okay, vectors are vehicles which will carry your DNA. Now this vectors, that is also a DNA, a recombinant, a vector and a gene of interest combines together to form a recombinant DNA. That recombinant DNA has to be transferred into the host cell by some methods, and that's the ones which are considered here, the first four ones. Now considering the other ones like vectorless or the direct gene transfer methods, you can take any DNA which is not being carried by a vector, and you can use techniques such as microinjection, particle bombardment, sonoporation, as well as liposome mediated gene transfer so that they enter into the host cell. We would be talking about each and every one of them in detail in the coming sessions. The first one is the transformation technique, and for the transformation to occur into E. coli cells, normally you have to make them competent. Competent means they should be capable of taking up DNA. So you prepare some host cells, you prepare the E. coli cells so that they become competent. And to make the E. coli cells competent, you give them a treatment with chemicals such as calcium chloride. And as a result, some pores are being created on the E. coli cells. And you take different aliquots, you chill them on the ice and you prepare different aliquots of the competent cells and you store them at -80. And when you need to transform your recombinant DNA onto the E. coli cells, you take the recombinant plasmid and you immerse it or inoculate it into one vial of the E. coli's competent cells, keep it on the ice for some time, and then you give it a heat shock at 42 degrees centigrade water bath, and after that you plate them onto the selective media. Now, since this recombinant DNA is containing an ampicillin resistance marker, you will use a agar plate which contains ampicillin. And after that, you will select the colonies which are having ampicillin resistance to find out whether they contain the particular gene of interest or the recombinant plasmid. So this is a common method which we use in the case of E. coli cells, the chemical transformation technique, where the host E. coli cells are being treated with calcium chloride and they become competent as a result of that. And the process of transformation, which mainly involves chilling on the ice so that the recombinant DNA enters into the cells, competent E. coli cells, and further on a heat shock is also being given and final plating is being done. Now you can do this transformation by electroporation techniques also. Sometimes the competent cells or the host cells which has to take the recombinant DNA would be tougher or would be reluctant, or the pores which are being formed by calcium chloride treatment would not be sufficient enough for it to pass a naked strand of DNA into it. So in such cases you use a technique such as the electroporation. Here also They have given you an example of E. coli. You can use it for E. coli also. But at the same time, you can use it for tougher cells like mycobacterium, depending upon who you are working on. So you take the cells, prepare competent cells as I had mentioned earlier. And after that, into the competent cells, your recombinant DNA is inserted, and then you give an electric shock onto it, and then wash it in a saline buffer, and then you immerse, plate it onto a selective media. So unlike the other step, in the previous step we had just used we had, after preparing the competent cells, we had given a what incubation. Just have a look. We had just incubated it on ice and then giving a heat shock. That was the transformation protocol that we used here. In electroporation, you are giving electric pulses onto the cells so that they get they get small pores, and as under the influence of the electric field, the DNA or the recombinant DNA which we are trying to transform here will enter into the competent cells, and finally they have been analyzed for their plating. I hope we can continue in the next class, and thank you for now, students.